Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma, and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our spring 2019 issue, titled Flow Cytometry: The Advanced Systems Overcoming Drug Discovery Pain Points. This article was written by Helen Stewart-Miller and Dr. Richard Massey of Biostrata. So now on to the main article, Flow Cytometry: The Advanced Systems Overcoming Drug Discovery Pain Points. Flow cytometry is an invaluable technique for the characterization of highly heterogeneous cell populations. Thanks to improvements in throughput, automation, and multi-channel detection capabilities, recent years have seen its use expand across a wide range of drug discovery applications. In this episode, we look at novel developments in flow cytometer reagent and instrument design and what these mean for drug discovery. Flow cytometry is an optical cell counting and characterization technique that enables large quantities of multi-parametric information to be rapidly collected on highly heterogeneous cell populations. Capable of analyzing many thousands of cells in a matter of seconds, flow cytometry offers formidable statistical power for screening applications, while its ability to simultaneously measure dozens of parameters on an individual cell basis gives it considerable advantages over bulk analysis techniques that average out important phenotypic differences. Improvements in speed, capacity, and usability over the past decade have seen flow cytometry used for a growing number of drug discovery applications, including biomarker discovery, ligand-binding assays, antibody screening, as well as target identification and validation. In large part, the adoption of flow cytometry across the length and breadth of the drug discovery pipeline has been driven by the ease of which systems can be integrated with automation platforms, making high-throughput screening applications increasingly cost-effective while helping pharma innovate faster. Despite these improvements, the need for pharmaceutical and biotechnology companies to do more with the same resources has put pressure on laboratories to derive additional value from their flow cytometry workflows. Fortunately, instrument and reagent vendors are meeting this need by developing solutions that make workflows more efficient and maximize the range of parameters that can be studied simultaneously. Pushing the limits of multi-parametric analysis. Flow cytometry measures how individual cells interact with light. The technique involves passing a thin stream of cells, often labeled using fluorescent dyes, known as fluorochromes, in front of one or more laser beams. Absorbed light is subsequently emitted by fluorescence at specific wavelengths, and the intensity of this light is measured using sophisticated optics and electronics. Because cells with specific phenotypic features generate differing emission spectra, valuable cell characterization information can be rapidly obtained. Ongoing advances in instrument design have resulted in the commercial availability of flow cytometers with an extensive range of laser and detector options. Together with improvements in the range of fluorescent dyes available for cell staining, this has led to an expansion in the number of wavelengths, or channels, that can be used to probe specific cell parameters. While these developments have enhanced the analytical power of multi-channel flow cytometry as a screening technique, this also necessitates the careful selection of laser, fluorochrome, and detector technologies to maximize the amount of useful information generated. One of the most important factors affecting the quality of flow cytometry data is the choice of channels used to measure specific cell parameters. However, this task is complicated by the fact that some chromophores fluoresce at very similar wavelengths, with varying degrees of intensity. If a detector assigned to pick up signals from a fluorochrome of interest detects fluorescence emission from a second fluorochrome, the physical overlap of emission spectra, known as spectral spillover, can make data interpretation challenging. This issue is even more acute if the fluorochrome of interest is weakly emitting or dim, making the effects of interference even more difficult to deconvolute. To address this issue, many instrument and reagent vendors are expanding and enhancing the range of detection channels available for flow cytometry experiments. One approach is to improve the quality of fluorochromes used for cell staining. Dr. Robert Balderas, Vice President Market Development for Biosciences at BD Biosciences, explains, "The limited availability of high-quality dyes with high extinction coefficients has been a major problem for laboratories, leading to suboptimal resolution of positive and negative cell populations. Our polymer dye technology platform gives researchers access to a broad range of fluorochromes, many of which are 20 times brighter than traditional dyes. This not only facilitates the use of new laser lines, it adds additional options to existing ones too." Another strategy to enhance the quality of flow cytometry measurements, especially when using dim fluorochromes, is to improve instrument sensitivity. One way this can be achieved is by using advanced detection technologies. Dr. Nancy Lee, Vice President of Engineering at ASEA Biosciences Incorporated, part of Agilent Technologies, explains, "Some of the most exciting advances in flow cytometry design relate to improvements in detector technology. Silicon photomultiplier (SIPM) technology, for example, offers high gain and high sensitivity for photodetection, which makes it possible to simultaneously measure a large number of fluorophores with excellent signal resolution. Our Novasight Quantian system is the first flow cytometer to adopt SIPMs for fluorescence channel detection." By increasing the number of viable channels that can be used for cell characterization and improving instrument sensitivity, cell biology can be studied in much greater detail, potentially putting more translationally relevant information in the hands of drug discovery scientists. These advances also allow the collection of more useful data from much smaller tissue samples. Not only is this more convenient for sample donors, it also allows laboratories to get more value from their existing workflows, helping to boost operational efficiency. As such, the latest advances in multichannel flow cytometry are poised to accelerate research and even improve success rates in a wide range of fields, including biomarker discovery and target identification. Realizing the full potential of automation. With throughput an important requirement for screening applications, manufacturers of flow cytometry platforms have devoted considerable resources to improving system capacity and speed in recent years. The commercial availability of instruments capable of processing large-scale micro 384 well plates at rates of tens of thousands of cells per second presents a wealth of opportunities for high-throughput screening applications. Moreover, the steady adoption of automation into flow cytometry workflows has led to more efficient processes that allow laboratories to work with much smaller volumes, boosting efficiency and measurement precision. Despite this, poor instrument flexibility has meant that manual instrument setup and switchover steps are typically still required, a time-consuming practice that prevents many organizations from fully realizing the full efficiency benefits of automation. Dr. Xiao Guo, Global Commercialization Product Manager at Bio-Rad Laboratories, explains, "A lack of flexibility when changing between sample formats has proven to be a major bottleneck in flow cytometry workflows, particularly for multi-user research labs that use a lot of different plate formats. To run plated samples, for example, incumbent systems typically require users to switch to an external plate loader and manually attach the plate sampling tubing to the sample injection port. These steps can be cumbersome and often require extra attention." Some instrument manufacturers, such as Bio-Rad Laboratories, are addressing this issue by developing systems with integrated sample loaders that can accommodate all widely used sample container formats. These systems reduce the need for manual changeover, freeing up scientists to work on other tasks. Additionally, the software solutions used to support these automated platforms are highly configurable, allowing laboratories to tailor their systems to their individual application needs. With these streamlined workflows, organizations engaged in drug discovery can benefit from more efficient processes, potentially accelerating successful outcomes. Commercially available flow cytometry systems. A broad range of flow cytometry systems is currently available on the market, and we'll now go through a selection. Firstly, Bio-Rad Laboratories' ZE5 cell analyzer is designed with throughput, speed, and flexibility in mind, and can be configured with up to 5 lasers and 30 detectors. With this high parameter setup, the system is capable of handling either high color panels or panels with minimal compensation. ZE5's fast fluidics and powerful electronics enable high-speed analysis of particles at 100,000 events per second without compromising data quality. Its innovative sample pump allows sampling from a volume as low as 2 microliters, which greatly saves both samples and reagents. The sample loading platform accommodates 40 or 24 tube racks, 96 and 384 hand deep well plates, and its smart sample handling features such as temperature control and vortexing capabilities are designed to maintain cell viability and prevent clogging. Furthermore, the system can be integrated into automated workflows and is ready to connect to laboratory information management systems and scheduling software to support new or existing automation solutions. ASEA Biosciences NovaCyte Quantian is a 4-laser benchtop flow cytometer that can be configured for up to 25 fluorescent channels to support the screening of large-scale panels. The system incorporates the most sensitive silicon photomultiplier detector technology on the market and offers a 7.2 log dynamic range, allowing both very dim and bright signals to be captured in the same analysis for the detection of rare events and low abundance phenotypic markers. Combined with the NovoSampler Q autosampler, the system boasts rapid analysis speed and allows a 96 well plate to be processed in less than 20 minutes. Automated after-sampling rinse ensures minimal carryover between samples, less than 0.1%. The system is compatible with all of the most widely used plate formats, from individual tube to 384 well plate, and can be integrated into automated workflows. The orbital shaker and barcode reader built in the NovoSampler Q provide a complete walk-away solution when integrated with a laboratory automation system, while an open application program interface allows for seamless operation with a scheduling software in a highly automated format. Fully automated calibration can get NovoSampler Q up and running within 5 minutes following installation. BD Biosciences offers a range of flow cytometers for research and clinical diagnostics applications. The BD LSR Fortessa X20 cell analyzer, for example, is a compact system built for high-performance multicolor analysis and can be configured with up to 5 lasers to facilitate the detection of as many as 20 parameters simultaneously. Thanks to its fully automated sample handling capabilities, the analyzer can process a 96 well plate in under 15 minutes, with less than 0.5% sample carryover. Its BD Accuray C6 Plus flow cytometer puts additional focus on usability and affordability, helping both novice and experienced users to benefit from flow cytometry. Incorporating 2 lasers, 2 light scatter detectors, and 4 fluorescence detectors, the system also features a user interface that is designed to be intuitive for non-experts. Beckman Coulter Life Sciences also offers a broad selection of systems for both routine and high-complexity flow cytometry applications. Its CytoFlex platform encompasses 3 compact instrument models designed to deliver optimal excitation and emission, minimize light loss, and maximize sensitivity. The most advanced system in this range, the CytoFlex LX, allows up to 6 lasers to be used, providing up to 21 fluorescence channels. The IntelliSite IQ Screener Plus platform is an integrated instrument, software, and reagent system that facilitates rapid, high-content, multiplexed analysis of cells and beads in suspension. 3 optical configurations are available, based on either 2 or 3 lasers, which permit the use of up to 15 detection channels. The system is designed to deliver insights quickly, with a detection rate of up to 35,000 events per second, and enhanced data visualization tools that support rapid interpretation of results. Conclusion. Improvements in flow cytometry reagents and instrumentation are helping to extend the analytical capabilities of this powerful technique. Recent advances in system flexibility, throughput, and multi-channel detection capacity are already helping scientists obtain more useful information from phenotypically diverse cell samples. As the insight that can be obtained from flow cytometry grows, its impact on existing and novel fields of drug discovery is set to expand. This article was written by Helen Stewart-Miller and Dr. Richard Massey. Helen Stewart-Miller is Director of PR Services, and Dr. Richard Massey is a science writing consultant at BioStrata, a life science specialist marketing agency. The company's growing team in Cambridge in the UK and Boston in the United States includes a significant number of people with deep scientific experience and knowledge. They offer a range of services from strategy, branding and message development, through to content creation, creative design, digital marketing, and public relations. If you've enjoyed this episode, then you can subscribe to Drug Discovery World free of charge by visiting our website at ddw-online.com, where you can also view all of our articles including references and images, and download the original PDFs. The links are in the show notes. If you've enjoyed the podcast, then do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter. Thank you for listening, and we'll hope to see you in our next episode.